Hello everyone, this is the module on evidence of evolution. Specifically, we're going to be talking about molecular biology. So the purpose of this is by the end of the module, you should be able to identify the two types of molecular evidence used to support the theory of evolution. So first, just to get started, we need to remember that we've just finished our unit on evolution and its mechanisms. And so just as a reminder, biologists can see evidence of evolution in every level of living systems. There is evidence of both past and present evolution. And the way that that's seen is through identifying and describing patterns in nature. So there's a variety of proof to support the theory of evolution, which includes fossils, anatomy and embryology, biogeography, direct observation, and molecular biology. For the purpose of this module, we're going to be focusing specifically on molecular biology, and then we will address the other variety of proof in the upcoming lessons. So first, the field of molecular biology is the field of biology that studies the composition, structure, and interactions of cellular molecules that carry out the biological processes essential for the cell's function and maintenance. And that's a handful. That's a lot. So what do we mean by this? What this really means is that all living things are made of chemicals. And molecular biology is specifically the branch of biology that studies how these chemicals or molecules interact and keep us alive. Specifically, the areas that are studied within molecular biology are primarily nucleic acid molecules and proteins. This is a very important area of study. This is what helps us understand disease, develop medicine, and in this instance, for the purpose of this module, specifically documenting evolutionary relationships. Now, just as a reminder, something that we've covered in the past and that is pertinent to this unit is that molecular biology is responsible for the central dogma. And that is the process by which the instructions in DNA are converted into a functional product. So that, just as a reminder, that is the taking of DNA, unzipping it, matching it, so transcribing it into RNA. That is then translated into a building chain of amino acids. Eventually, those amino acids fold into a variety of shapes, and that is what we call a protein. So we have a couple of definitions. You are going to have these here so that you can reference as you need to, and so that you can watch this instruction and as well as the other instructions repeatedly. But to start, just as a reminder, these are things that we've covered in the past, but molecules are the smallest particle of a substance that has all of the physical and chemical properties of that substance. Molecules are made up of one or more atoms. So if you look over to the right side, we have an atom which represents a single neutral particle. In this case, it's an oxygen atom. When you combine that oxygen atom with two hydrogen atoms, that forms a water molecule. And the definition of a molecule is neutral particles made of two or more atoms bonded together. Biological molecules, also known as biomolecules, can be made of many thousands of atoms. Molecules that are produced by living organisms sorry about that. Technical difficulties. As I was saying, biomolecules are molecules that are produced by living organisms. The major groups include carbohydrates, which is essentially they're just sugars, lipids, which are fats, nucleic acids, and those are things like DNA and RNA, and proteins. Nucleic acid molecules are naturally occurring chemical compounds that carry information for cellular life. This includes genetic material. And so that, as a reminder, those are RNA and DNA molecules. It's very difficult and expensive to sequence or to sort of map out and name an entire genome. And as a reminder, a genome is the complete set of genes or genetic material present in a cell or organism. So instead, what biologists do, instead of trying to map out and match up entire genomes of organisms, biologists focus on a very specific set of genes which are specific and important to the organism being studied. So as an example, microbial evolution is traced through genes called 16S rRNA. Mammalian evolution is commonly traced through mitochondrial DNA, and which is abbreviated as MT, lowercase mt DNA, and that is called, this particular one that we use to trace is cytochrome B. Proteins are another biomolecule that can be used to trace evolution through the molecular units. So proteins are made of amino acids, which again are molecules, and amino acids are the smallest unit of a protein. So if a protein breaks down completely or it's degraded completely, you are still left with all of those amino acids. Proteins and amino acids combined are known as the building blocks of life. Because proteins are sequences of amino acids copied directly from translated chains of RNA, as a reminder, that's what is described in the central dogma, proteins can also be used to trace an evolutionary lineage. A lineage is a sequence of evolved species. Proteins also serve as a variety of functions in a living system. Sometimes they are enzymes, which they help catalyze reactions or make reactions happen more readily. They can be structural material as keratin. They can provide transport as hemoglobin in the blood. They can provide immunity in the form of antibodies, and they're important for regulation. Differences in proteins come down to the amino acid composition, sequence, location, function, and spatial configuration. So again, that is all dictated by the genetic material translated into amino acids. The amino acids in a protein are determined by the genes coding for them. As a reminder, that's a central dogma. So as we look off to the right, the top picture is another image describing the central dogma where double-stranded DNA is separated and then copied into a single strand of RNA. That mRNA copy is then translated into a chain or sequence of amino acids. Those amino acids are then folded into a variety of structures. The primary structure is just you can imagine it as just a straight train as it's building that eventually folds into a secondary structure. But where they're actually primarily used are in the tertiary or third structures or quaternary structures, or fourth structure. So you can see that they are folded in very complicated ways, and that folding, as well as the sequencing, determines what kind of protein it is, what that function of that protein is. So how does all of this information help us understand evolution? So the genetic sequence, or the nucleic acids, and amino acid sequences, or the proteins, can be compared from one organism to the next. The more similar the sequence, the more closely related the organism. The more dissimilar the sequence, the less closely related to the organism. So if you look down below at this chart, or this diagram, what we're looking at is a phylogenetic tree, which is something that we're going to talk about later. This is just a graphic way to compare living organisms and to explain when they diverged or split from each other. If you look at these sequences and you align them, you can see that the human and chimpanzee have similar alliances. So you can see that the sequences are happening in conjunction at the same time gap sequences are happening in conjunction. That similarity tells us that there is a common ancestor and that we haven't diverged from them comparatively as long ago as we have, say, from gorillas, and even longer from orangutans. And you can see that as you go down the chart, that these sequences and gaps start to become more different. We can compare genetic sequences because genes are heritable or passed down from one generation to the next. Differences in sequences can tell us when organisms evolved away from each other. What biologists also do is they use a technique called a molecular clock. A molecular clock is used to determine how closely related two species are by calculating the number of differences in a genetic or protein sequence. The fewer the differences, the less time the species have diverged or split and evolved into new species. What scientists actually have to consider is the specific organism that they're studying when they are determining the molecular clock. And the reason is because they need to count or understand when mutations are happening in genetic material. Mutations are the driving force behind evolution. So a mutation in genetic material must have a standard rate when comparing from one organism to the next. Rates specific to the organisms are the area of study. That's really the importance here, that rate of mutation. This information is then used to determine how long ago species diverged. And if you take all of this information into consideration, these are the patterns that help us see evolution. If you look over to the right, this again is a graphic display of a phylogenetic tree. It's again something that we're going to talk about again in upcoming units, but it's just a way to illustrate relatedness and the amount of time between when these organisms were related to each other and when they diverged. So this one in particular is microbial. This is bacteria. So you can see these two types of plasmodiums at the top, they diverged 100 million years ago. And again, scientists are able to calculate that by using a standard rate of mutation for microbes and using a molecular clock. This is a larger picture of a molecular clock, again illustrated in phylogeny. If you look along the x-axis, you can see that the bottom row is specific to genes. That's specific to the nucleic acids. So again, 16S RNA is used for bacteria, and we can see that in the beginning of this chart, this is specific to microorganisms. As we progress along the chart, we get more advanced as we go. If you look up the second x-axis, that is proteins. And then a little bit higher and beyond the scope of this course is neutral non-coding DNA. I'm not sure we're going to get to that, but it is something that you can look up and something that I would be happy to help you look up if you're interested in that. If you look along the y-axis, you can see that this chart is in millions of years, because evolution, while it can happen from one generation to the next, the process of speciation is long-term.